Hello and welcome everybody to today's edition of the 3D Regulatory Genome, Enhanced Genomics virtual seminar series. And I'm delighted and excited to welcome Professor since this year, Professor Alvaro Rada-Iglesias, as our speaker. Alvaro has obtained his PhD in Uppsala, in Sweden, where he worked in the laboratory of Professor Claes Vadelius, and then joined, supported by an EMBO long-term fellowship, the lab of Professor Johanna Wysoka at Stanford University, where he made key contributions to our understanding of enhancers and different classes of enhancers, histone modifications that characterize these enhancers. In 2013, Alvaro was then recruited to the University of Cologne as a junior research group leader at the Center of Molecular Medicine. And this is where his independent career started. And since 5 years later, then in July 2018, he was recruited as a principal investigator by the University of Cantabria, and where he is still today at his group at the Institute of Biomedicine and Biotechnology of Cantabria. And as he became an associate professor in 2020 and a full professor just recently in 2025. And his work has been recognized by several awards, including the EMBO Young Investigator Award, the Jose Luis Gomez Scarmetta Award, and an ESC Consolidator grant. And I'm very excited. Sorry, a few more housekeeping things. So this talk will be recorded, except for the discussion. So we keep that a bit more kind of lighter. And if you have any questions, please put them in the Q&A box in the beginning, and I'll read those out and then I'll pass them on to Alvaro. Yeah, and with that, I'll pass over to Alvaro. Very excited and really looking forward to your seminar.
Thank you, Stefan. So I'm gonna share my screen. Hopefully everything is gonna be fine. Give me a second. Right, you see it, right?
I can see it. Yeah.
All right, perfect. So yeah, thanks, Stefan, for the kind introduction, and also for the opportunity to present some very recent work from the lab, in which basically we are using what we consider synthetic engineering approaches in order to gain some new mechanistic insights into long-range gene regulation. So let's see. It seems like I have some problem to move no. So during vertebrate embryogenesis, the establishment of cell type specific gene expression programs is largely dependent on regulatory activity of a broad set of distal cis-regulatory elements that we typically refer to as enhancers. According to classical and operational definitions, enhancers are basically short DNA sequences containing clusters of transcription factor binding sites, and that can positively control the expression of their target genes in a distance and orientation independent manner. In other words, the enhancers can, in principle, control the expression of genes that are located at very long linear distances, and they can be located either upstream, downstream, or even within the genes they regulate. However, this classical view, according to which enhancers can control the expression of the target genes regardless of the linear distance separating them, has been recently challenged by a number of studies, including some beautiful work from Luca Giorgetti's lab using enhancer mobilization assays. According to these studies, it's been shown that actually increasing the linear distance between enhancers and target promoters can actually diminish gene expression. And moreover, in another recent work also from the Giorgetti lab, it was shown that actually increasing these linear distances separating enhancers and promoters not only reduces gene expression, but also burst frequency and transcriptional precision, which has led to the suggestion that long-range enhancers might be less precise than short-range ones. However, these suggestions are difficult to reconcile with the fact that major developmental genes, which typically display very precise and robust expression patterns, are often regulated by very long range or even ultra long range enhancers that sometimes can be located more than 1 megabase away from their target gene, as illustrated here by the CRS enhancer, which is essential for proper expression of the sonic hedgehog gene during limb development. Therefore, there must be certain mechanisms that allow long-range enhancers to confer their target developmental genes with very precise, robust, and specific expression patterns, as illustrated here for several developmental genes that are expressed during either mouse brain or mouse limb development. Moreover, these long-range enhancers need to confer this very precise and robust expression, despite the fact that transcription is considered to be a rather noisy, stochastic, and bursty process. Previous studies have suggested that different types of enhancers, including, for example, super enhancers, CTCF-associated enhancers, as well as CpG-rich enhancers, can be particularly suitable and important in conferring this type of precise and robust transcription that developmental genes and cell identity genes seem to require. Super enhancers are basically clusters of several enhancers that are close to each other in linear space. And it's been proposed that these type of enhancer clusters can drive a strong and robust gene expression by facilitating the formation of transcriptional condensates through liquid-liquid phase separation. However, it's still heavily debated whether this type of enhancers truly display emergent regulatory properties or whether they rather reflect the addition of several typical enhancers. On the other hand, there has been also studies indicating that intra-TAT CTCF sites that are associated with both distal enhancers and promoter regions can actually facilitate enhancer-promoter communication and thereby increase target gene expression. However, when considering together these studies where this type of CTCF sites have been investigated, it turns out that this type of intra-TAT CTCF site might be particularly important for the activity of ultra-long-range enhancers, while short-range enhancers might be less dependent on this type of CTCF sites facilitating enhancer-promoter communication. Last but not least, our lab previously described that CpG islands that are associated with both distal enhancers and the promoter regions of developmental genes can act as tethering elements that increase the physical and functional communication between distal enhancers and developmental genes, thereby increasing the expression of this type of genes and also transcriptional precision. However, it's important to consider that these previous studies in which these different types of enhancers have been investigated have been performed under quite different experimental conditions, potentially introducing multiple confounding factors such as different cell types, differences in promoter type, differences in enhancer composition, etc., that together can make it difficult to discern the unique or distinctive regulatory properties of each of these enhancer types. In this regard, we hypothesized that the experimental strategy that we previously used to dissect the regulatory logic of CpG-rich enhancers could be quite insightful. Basically, based on previous work on this type of enhancers, we hypothesized that CpG-rich enhancers display a unique modular composition consisting of a core enhancer that contains a cluster of transcription factor binding sites and a nearby CpG island. We also hypothesized that under this modular composition, the core enhancer could confer the cis activation capacity, while the CpG island could bestow permissive chromatin and topological features that can boost the activity of the enhancer and thereby increase transcriptional precision. In order to test this hypothesis and thereby dissect the regulatory logic of CpG-rich enhancers, we implemented a synthetic engineering approach whereby we generated transgenic mouse CSL lines in which the components of CpG-rich enhancer that we previously characterized and that gets specifically activated in neural progenitors were introduced either alone or in combination at 100 kb from the promoter regions of 2 different genes. On the one hand, we have GATA6, which is a typical developmental gene whose promoter contains a large cluster of CpG islands. And on the other, we have GRIA1, a tissue-specific gene whose promoter does not contain CpG islands. Importantly, both GATA6 and GRIA1 are inactive in both ES cells as well as in neural progenitors, the cell type in which our model enhancer gets specifically activated. And therefore, both GATA6 and GRIA1 regulatory domains are devoid of active enhancers in these 2 cell types. This is quite important because basically, upon differentiation of our transgenic cell lines into neural progenitors, any changes in the expression of either GATA6 or GRIA1 would be in principle solely attributed to the inserted enhancer components. So once these transgenic cell lines were obtained, we differentiated them into neural progenitors and then measured both GATA6 and GRIA1 expression. Regarding GATA6, what we found, first of all, was that although the core enhancer alone already resulted in the induction of GATA6 in neural progenitors, such induction was strongly increased when the enhancer was combined with a CpG island. Moreover, we also saw that the CpG island alone did not induce GATA6 expression, indicating that the CpG island by itself does not have cis-regulatory activity. On the other hand, regarding the GRIA1 cell lines, what we found is that, in contrast to what I showed you for GATA6, upon differentiation into neural progenitors, none of the enhancer combinations that we tested resulted in GRIA1 expression. So these results, first of all, indicate that CpG islands can indeed boost the regulatory activity of a nearby CpG island, but that they specifically do so in the context of developmental genes whose promoter also contains CpG islands and that therefore seem to be particularly responsive to this type of distal enhancers. Next, in order to gain some mechanistic insights into how the CpG islands can boost the activity of the nearby enhancer, we performed several experiments, including 4-6 assays in our different transgenic cell lines. In the case of the GATA6 cell lines, we use as viewpoints in these 4-6 experiments either the GATA6 promoter region or the insertion site of our synthetic enhancers. And importantly, strong enhancer promoter contacts were only observed in cells containing the core enhancer together with a CpG island, therefore in agreement with the strong induction of GATA6 in those same cells. In contrast, when similar 4-6 experiments were performed in the GRIA1 cell lines, in this case, we could not observe clear or strong enhancer promoter contacts for any of the investigated enhancer combinations, therefore also in agreement with the lack of GRIA1 expression in, upon differentiation of these cell lines into neural progenitors. So together with many additional data and experiments that I don't have time to show you, in this work, we basically uncover a novel role for CpG islands as tethering elements that increase the physical and functional communication between distal enhancers and developmental genes. Importantly, these functional mechanistic properties that I just showed you for CpG islands seem to be quite similar to what was recently described by Mike Levine's lab in Drosophila, suggesting that this type of tethering elements are probably evolutionarily conserved. So based on this previous work, we hypothesized that a similar synthetic engine approach to the one I just described could actually allow us to compare the regulatory properties of super enhancers and CTCF-associated enhancers under almost identical conditions, and therefore gain mechanistic insights into the regulatory properties of these different enhancer types and how they robustly control the expression of their target genes. So briefly, what we decided to do is to adapt our synthetic engine approach to these new types of synthetic enhancers. Basically, we have been using the same core enhancer that we used in our previous study, and that is associated endogenously with the SOX1 gene, and that again gets specifically active in neural progenitors. So using this core enhancer, we first of all combine it with a CTCF site, and then we also create another synthetic enhancer basically containing 4 tandem copies of our core enhancer, and we consider this to basically represent an enhancer cluster that could somehow resemble the genetic properties of super enhancers. Once these synthetic enhancer constructs were obtained, we again inserted them at 100 kb downstream from the GATA6 gene and generated transgenic mouse cell lines. I would like to mention that regarding the enhancer associated with a CTCF site, the CTCF site that we used actually overlaps a strong CTCF peak located close to the Sonic Hedgehog gene. And importantly, this CTCF site was inserted in a convergent orientation with respect to endogenous CTCF site located at the GATA6 promoter region, as this theoretically should facilitate the communication between the GATA6 promoter and our synthetic enhancer. So once we generated these transgenic cell lines, what we did was, as I mentioned before, to differentiate it into neural progenitors and then analyze these cells with a battery of methodologies in order to systematically compare the regulatory properties of the different types of synthetic enhancers. But before we did that, we wanted to make sure that our synthetic enhancers display the expected molecular features. Regarding the enhancer associated with CTCF binding site, what we did was to perform ChIP-seq experiments for CTCF and the cohesin subunit RAD21. And as you can see in these ChIP-seq tracks, both in mouse cells as well as upon differentiation into neural progenitors, the addition of the CTCF site to our core enhancer resulted in the strong recruitment of CTCF and also importantly in the stalling of cohesin, as illustrated by these strong RAD21 peaks that we observe both in S cells as well as in neural progenitors. On the other hand, regarding our 4X enhancer cluster, what we did here was to perform ChIP-seq experiments for MET26, one of the mediator subunits, as one of the main molecular features of super enhancers that they recruit high levels of mediator. And in agreement with these features, what we found is that our 4X enhancer cluster recruited almost 5 times more mediator than the core enhancer alone. So being reassured that our synthetic enhancers display at least some of the expected molecular features, we then went ahead and differentiated them into neural progenitors and first of all measured GATA6 expression by RT-qPCR. Somehow to our surprise, first of all, we noticed that the addition of the CTCF site to the core enhancer did not have any major impact on GATA6 expression in comparison to the core enhancer alone. In contrast, the 4X enhancer cluster displays a strong and synergistic increase in GATA6 expression in comparison to the core enhancer alone, since basically, although this enhancer cluster has only 4 more copies than the core enhancer, it actually led to a 12-fold increase in GATA6 expression in comparison to this core enhancer. So this initial set of experiments indicate that in agreement with previous reports regarding the regulatory properties of super enhancers, our 4X enhancer cluster display emergent regulatory properties and a strong regulatory capacity. As probably most of you know, transcription does not occur in a continuous manner, but rather in the form of bursts, which can be defined by several parameters, including burst amplitude and burst duration, which together define the burst size, as well as burst frequency. Importantly, previous studies have demonstrated that during gene transcription, enhancers are mostly in charge of controlling burst frequency, while promoters seem to preferentially control burst size. So we wonder whether our synthetic enhancers could somehow differ in how they control these different bursting parameters. This type of bursting parameters can be measured or estimated using different methodologies, including single molecule RNA-FIS. In this type of assays, one needs to use fluorescent probes against intronic and exonic sequences of a gene of interest. And once these intronic and exonic signals are acquired, the next step is to identify those foci in which the intronic and exonic signals overlap, and that basically will correspond to those locations in which the target gene is undergoing nascent transcription. Once these foci are identified, burst size can be basically measured as the intensity of the intronic signals at those foci, while the burst fraction, which is correlated with burst frequency, can be calculated by counting the overall number of foci in which the intronic and exonic signals overlap in the investigated soil population, and this basically allows them to calculate the number of transcribing alleles per cell. So basically what we did was to differentiate our transgenic cell lines into neural progenitors and then perform single molecule RNA-Fis experiments in those cells for GATA6. So then we identify all those foci in which the intronic and exonic signals overlap, and this allowed us to calculate both bar size and bar fraction. And in agreement with the RT-qPCR data that I just showed you, first of all, what we observed is that the addition of the CTCF site to the core enhancer did not seem to have any major impact on either bar fraction or bar size in comparison to the core enhancer alone. In contrast, for the 4X enhancer cluster, we again observe a synergistic increase in bar fraction, but also a more moderate, but nevertheless non-negligible effect on bar size. Actually, if we consider together the effect that the enhancer cluster has on both bar size and bar fraction, together they seem to nicely explain the overall increase in GATA6 expression that we measure in bulk by RT-qPCR. So these single molecule RNA-Fis experiments indicate that in agreement with previous reports regarding how enhancers control burst parameters, basically our data indicates that the enhancer cluster preferentially increases GATA6 expression by controlling or increasing bar fraction, but we also observe effect on bar size, which based on some recent reports, we speculate could be related to a mild increase in bars duration. Moreover, there has been a number of studies indicating that enhancers control bar fraction by actually controlling and increasing enhancer promoter contact frequency. So we wonder whether our enhancer cluster could actually synergistically increase GATA6 expression and bars fraction by actually contacting the GATA6 promoter with it. In order to test this, we decided to perform Capture-C experiments upon differentiation of our transgenic cell lines into neural progenitors and using as viewpoints either the GATA6 promoter region or the enhancer insertion sites. And we did that in collaboration with Marieke Odela's lab. Moreover, this type of Capture-C assays will also allow us to address whether despite the fact the addition of the CTCF site to the core enhancer did not have any major impact on GATA6 expression, it could nevertheless perhaps have an impact on the 3D chromatin organization of the GATA6 domain. So I'm going to first show you the Capture-C experiments using the GATA6 promoter as a viewpoint. And this is shown either as individual tracks for each cell line, as well as subtraction tracks relative to the wild-type signals. So upon inspection analysis of these Capture-C datasets, one of the first things we notice is that the core enhancer alone was already able to establish clear contacts with the GATA6 promoter region. And more interestingly, we also notice that the frequency of those enhancer promoter contacts were actually very similar to the ones observed when the core enhancer was combined with a CTCF site or if we inserted a 4X enhancer cluster. Regarding the cells with the core enhancer together with a CTCF site, the fact that we didn't observe major changes in enhancer promoter contact frequency were actually in agreement with the lack of effect on GATA6 expression. However, upon closer inspection of the data, especially if one looks at the subtraction tags, we notice that the addition of the CTCF site seems to have an insulatory effect, since basically we found that the contact frequency between the GATA6 promoter region and the regions located downstream of the inserted CTCF site seem to be diminished in these cells containing the CTCF site, while the contact frequency of the promoter with regions located immediately upstream of the CTCF site seemed to increase. Actually, this insulatory effect became even more evident when looking at the Capture-C data that we generated now using the insertion sites as viewpoints. And here, what you can see is that although we now observe a mild increase in enhancer promoter contact frequency, what is very obvious is that upon an insertion of the CTCF site, the contact frequency, now in this case of the enhancer, with regions located downstream of the CTCF site actually were clearly reduced, while at the same time there is a clear increase in the contact frequency of the enhancer with regions located towards the 5' end of the GATA6 domain, including clear interactions with CTCF sites that overlap the 5' boundary of the GATA6 domain. So these results regarding the cells in which we inserted a CTCF site together with a core enhancer indicates that the CTCF site does not strongly affect enhancer promoter contact frequency or GATA6 expression, but instead leads to the formation of a smaller regulatory domain that we can perhaps consider to represent a subtat or chromatin hub. And this is in perfect agreement with some recent data also from Marieke's lab using CTCF diglones. On the other hand, regarding the cells with the 4X enhancer cluster, as I mentioned before, using the GATA6 promoter as a viewpoint, but you can also see here when using the insertion site as a viewpoint, we did not observe any major changes in the contact frequency between the enhancers and the GATA6 promoter region in comparison to cells having the core enhancer alone, indicating that the strong increase in GATA6 expression driven by the 4X enhancer cluster cannot be explained by changes in enhancer promoter contact frequency, and instead led us to speculate that perhaps the 4X enhancer cluster is actually increasing GATA6 expression by increasing the proportion of such contacts that are actually transcriptionally productive. One possible mechanism that could explain how the enhancer cluster synergistically increases GATA6 expression without having a major effect on enhancer promoter contact frequency is by facilitating the formation of transcriptional condensates. As I mentioned earlier, super enhancers are basically considered to be clusters of several enhancers that are close to each other in linear space. And these enhancer clusters can recruit high levels of transcription factors and different coactivators such as mediator or BRD4, which are rich in intrinsically disordered regions. And this ultimately in turn can lead to the formation of transcriptional condensates through liquid-liquid phase separation. Importantly, some recent studies indicate that this type of transcriptional condensates are rather large entities with more than 500 nanometers in diameter, which is actually larger than the distance that enhancer promoter contacts seem to entail, which normally it's believed to be around 200 nanometers or less, which means that basically if our 4X enhancer cluster is indeed facilitating the formation of this type of condensates, the enhancer cluster and the GATA6 promoter can perhaps reside in one of these condensates without really requiring to establish close physical contacts. So in agreement with the possibility that our enhancer cluster could drive the formation of these transcriptional condensates, when we performed ChIP-seq experiments against several coactivators, not only MET26, but also CDK9, P300, BRD4, which is not shown here, we actually observed that in general, the 4X enhancer led to an either additive or more often subadditive increase in the recruitment of pretty much every coactivator that we checked in comparison to the core enhancer alone, which will agree with, as I said, this possibility that the 4X enhancer could drive the formation of condensates. The formation of these condensates by super enhancers has been previously addressed, at least in vivo, by combining RNA-Fis experiments against genes controlled by super enhancers, together with immunofluorescent assays for coactivators such as BRD4 or mediator. Importantly, what was shown in this study is that upon averaging the coactivator signals around the RNA-Fis foci of those genes controlled by super enhancers, it turns out that these genes more often overlap with coactivator condensates than one would expect by chance, or also in comparison with genes regulated by typical enhancers. However, in these previous studies, it was not clear or was not directly addressed whether this condensate formation was really causative of transcriptional activity, or in other words, whether there was a positive correlation between condensate formation and transcriptional activity. So in order to address whether our enhancer cluster could indeed be driving transcriptional condensate formation, we performed this kind of experiments that I just described. Basically, we differentiated our transgenic cell lines into neural progenitors and then combined single molecular RNA feeds against GATA6 and immunofluorescence assays for BRD4. And indeed, upon averaging the BRD4 signals at around hundreds of GATA6 RNA feeds foci, we actually observed that in cells containing the 4X enhancer cluster, those GATA6 RNA feeds foci seem to overlap with higher levels of BRD4 that potentially represent transcriptional condensates. Nevertheless, upon direct comparison of the BRD4 signals and the GATA6 RNA feed signals at the analyzed GATA6 RNA feed foci, we actually observe a very weak positive correlation between both signals in cells having either the core enhancer alone or the 4X enhancer cluster, which basically suggests that the strong regulatory capacity of our 4X enhancer cluster and the synergistic increase in GATA6 expression that we observe in those cells cannot be easily explained by the formation of transcriptional condensates. In addition to the formation of transcriptional condensates, those co-activators that display higher recruitment to our 4X enhancer cluster, as well as to super enhancers in general, have been previously implicated in the control of several steps along the transcriptional cycle, including pre-initiation complex formation and initiation, as well as RNA pol-2 post-release and elongation. In order to address whether our 4X enhancer cluster could preferentially control some of these transcriptional regulatory steps in comparison to the core enhancer alone, we decided to perform ChIP-seq experiments against the hypophosphorylated form of RNA pol-2, which should serve as a marker of pre-initiation complex formation and initiation, as well as ChIP-seq experiments for total RNA pol-2 that should allow us to detect elongating RNA pol-2. So upon analysis of these ChIP-seq profiles, what we observe, first of all, is that both the core enhancer and the enhancer cluster led to a strong recruitment of hypophosphorylated RNA pol-2 around the GATA6 promoter region. In contrast, when measuring total RNA pol-2 within the GATA6 gene body, what we found is that the 4X enhancer cluster synergistically increased the amount of elongating pol-2 within GATA6, which basically indicates that the 4X enhancer cluster is not increasing GATA6 expression by dramatic increasing the amount of pre-initiation, complex formation, or initiation, but rather by facilitating RNA pol-2 post-release and therefore transcriptional elongation. In agreement with this notion, we then calculated the posing index for GATA6 in using our ChIP-seq profiles, and indeed we found that the 4X enhancer cluster led to a significant reduction in this posing index, again indicating that the 4X enhancer cluster could actually facilitate RNA2 post-release. So altogether, these ChIP-seq profiles indicate that our core enhancer, and perhaps according to previous studies, enhancers in general, preferentially regulate gene expression at the level of transcription initiation. In contrast, our data indicates that enhancer clustering can increase target gene expression preferentially by facilitating RNA pol-2 post-release and thereby transcriptional elongation, which has been, for example, previously proposed in Drosophila for at least some super enhancers. So finally, in order to test whether these emergent regulatory properties and the associated mechanisms that we described for the 4X enhancer cluster could be somehow extended to other developmental loci, we decided to generate additional transgenic cell lines in which the same synthetic enhancers were now inserted 25 kb downstream of SOC7, another developmental gene that, similar to GATA6, contains a large CpG island cluster around its promoter region, and also similar to GATA6, this gene is inactive in both ES cells and in neural progenitors. So once we generated this additional set of transgenic cell lines, we basically differentiated them into neural progenitors, and then used several of the methods that I just mentioned in these cells in order to, again, compare the core enhancer and the 4X enhancer regulatory properties. So first of all, and importantly, what we found is that upon differentiation into neural progenitors, the 4X enhancer cluster strongly and synergistically increased SOC7 expression in neural progenitors in comparison to the core enhancer alone. In this case, we actually observe an even stronger 45-fold increase in GADA6 expression when comparing the enhancer cluster and the core enhancer. Next, we also performed single molecular RNA-FIS experiments for SOC7 upon differentiation of the transgenic cell lines into neural progenitors. And as I showed you for GADA6, here again, what we found is that the 4X enhancer cluster synergistically increased Barr's fraction, while again having a milder but non-negligible effect on Barr's size. And finally, we also performed capture-C experiments upon differentiation of these SOC7 cell lines into neural progenitors using the SOC7 promoter region as a viewpoint. And in this case, what we found is that the enhancer cluster display a mild increase in enhancer promoter contact frequency, but that nevertheless is much lower than the 45-fold increase in SOC7 expression driven by the enhancer cluster. So altogether, this set of results in the SOC7 cell lines further support our idea that the emergent regulatory properties of enhancer clustering could preferentially entail an increase in the proportion of productive enhancer promoter contacts rather than an increase in enhancer promoter contact frequency. So to sum up, I hope I convince you that using synthetic engineering or synthetic biology approaches can be quite useful in gaining mechanistic insights into long-range gene regulation. In this particular work that I showed you today, first of all, we've shown that intra-TAT CTCF sites do not seem to universally facilitate enhancer promoter contacts or thereby target gene expression. But instead, what our data suggests is that a more general role of these intra-CTCF sites is to facilitate the formation of chromatin hubs or subtats within larger TAT domains. And we speculate that the regulatory importance of these intra-TAT CTCF sites and the associated chromatin hubs that they might form might be particularly relevant in the context of ultra-long range enhancers separated by very long linear distances from their target genes. More importantly, at least in our opinion, our work also revealed that the strong regulatory activity of enhancer clusters, which we believe will also apply to super enhancers, is unlikely to be explained by any major changes in enhancer promoter contact frequency or the formation of transcriptional condensates. Instead, our data suggests that the emergent regulatory properties of enhancer clusters preferentially entail a strong increase in RNA pol-2 post-release and elongation, and thus in the proportion of transcriptionally productive enhancer promoter contacts. So with this, I will finish by acknowledging the people that actually did the work. And actually, I would like to especially emphasize the work of one of the postdocs in my lab, Andy Carroll, that actually generated all the cell lines and also analyzed most of the data that I showed you today. We also got help from Marieke Oudelar's lab with some of the capture-C experiments. And finally, I would also like to thank our funding sources and all of you for your attention. Thank you.
Thank you very much.